I'm a PhD student with Dr. Jim Reed. I look at specifically the evolution of the carbon fixing enzyme in the C4 process. The C4 process is an adaptation grasses have made to improve the photosynthetic efficiency of the species. So what it does is, rather than preventing oxygen getting to the active site of an enzyme that makes sugar, it prevents it completely by pumping in carbon dioxide in a 4-carbon system. Now, it did this over 60 different times in nature by recruiting the enzymes for this cycle, the C4 cycle, from other roles within the plant, and then improving the kinetics for the role of C4 photosynthesis. So we're trying to understand what kinetics were chosen, why they're chosen, and how they were selected. And these are, it should be noted, the enzymes before and after evolution, as it were, basically look exactly the same in 3D space. So the evolution is a result of single amino acid changes. And interestingly, rice is not a part of a family that can evolve this. So what we can do is we can work out how these changes occurred and then induce them in rice, so that rice, which is very inefficient, it lives in a very water heavy environment, can become more efficient and produce more rice, because if you give it enough efficiency, it will just indefinitely produce a large amount of rice. So the work is part of the greater C4 rice project, which is, the aim is to engineer the C4 process into rice. So this is a collaboration between chemists, biologists, biological engineers, chemical engineers, geneticists in the Philippines and across the world, based in the Philippines, mind you, to change the genetics of rice and engineer the enzymes to fully induce the C4 process into rice. And it looks like it's coming along quite well. In order to look at the kinetics of an enzyme, I actually have to make the enzyme. So what we do is we put the piece of DNA that encodes the enzyme in bacteria, grow the bacteria till we've got enough, then use a chemical messenger to tell it to make the specific protein. When we've got enough of that, we then freeze it, and then we release once we need it, then we defrost it and suspend it in a buffer, and then sonicate it so that we release the actual enzyme from inside the bacteria. We then purify that with a series of processes. Now, the most common one, and the one that we use, is called a histag, where you change the DNA structure of the enzyme. You have your enzyme code there, and just before it, you put the code for 6 histidines, and this has histidine amino acids have a high affinity for nickel. So our protein sticks to nickel quite easily, and then everything else washes down a column, and then we get a basically pure enzyme. There's a couple of polishing steps afterwards. It's 1 mil histidine trap, and what that does is it's got nickel bound to plastic beads, which then my protein binds, and then the other proteins in a bacteria then just do not bind and wash straight through, and hopefully that will get pure enzyme. And behind you is the active purification system. This works exactly the same, except you put the column in it, you connect it up with the tubes, and then it automatically pumps all the solution through for you, rather than you having to literally put it in. And you fill your enzyme in this tube and it automatically pumps it in, so that way there is no room for human error, as long as you programmed it correctly. But it's a very nice machine. It stays at 4 degrees for the maximum stability of the protein. So we use columns like these, and this one we made ourselves, it's a desalting column, and yeah, that's how we purify protein. It is a big difference learning all the genetic stuff, but it is well worth it, and it's not that difficult, because chemistry is very good at teaching you how to learn. Yeah. So I work on the enzyme PepC, and PepC binds bicarbonate as well as phosphoenol pyruvate. So a lot of work has gone into finding out how the enzyme evolved by binding phosphoenol pyruvate. Little work has been looked into bicarbonate. So I've set up a system to reduce background bicarbonate, and that involves sparging it with the assay buffers with sparging with nitrogen, and that's what the gas systems I've got set up is. So I started in my undergrad. I was very interested in medicinal chemistry, but actually that wasn't very interesting to me, and I started looking at how medicines interact with biology on an enzymatic scale, and then kind of got rid of the organic stuff altogether and was just more interested in how enzymes work. And I did my undergraduate project on insulin, and that was quite productive, and so that's why I'm here now, because I looked at how insulin changes with single amino acid changes.